So I got cut off from my last video, but we're going to just continue here. Uh, the rest of the day here is. And so I will show my greatness and my holiness, and I will make myself known in the sight of many nations. They will know that I am the Lord. Ezekiel 30:22. And we're going to go into hypersensitivity reactions. Type 1, hypersensitivity. We said there were two types, an immediate type and a late one. Um, so in the very first reaction, you have three stages. The first stage is pre-sensitization, which is when you have prior exposure to the allergen. So here, in a previous exposure to an allergen, such as peanuts or bee venom, the body makes IgE antibodies specific to that allergen. In this case, we're going to use bee sting. Okay, so in a previous exposure to a bee sting, the body will make IgE antibodies to that bee sting. These IgE antibodies bind to the FcRI receptors on mast cells and basophils. This is called sensitization. Next stage is re-exposure. When the same allergen, like the bee sting, enters the body again, the allergen binds to two or more IgE molecules on the surface of the mast cell. This cross-links the IgE antibodies. Third step is mast cell activation. So the cross-linking of the IgE antibodies on top of the mast cell causes the mast cell to degranulate, releasing histamine, tryptase, and leukotrienes and prostaglandins into the blood. Immediately you will see symptoms within minutes. You will see anaphylaxis, which is hypotension, bronchospasm, hives, angioedema, shock. So basically, the body produces IgE when exposed to an allergen. And then these IgEs go and bind to the mast cells, right? They go and bind to the mast cells after they are produced. This part is known as sensitization. And then later, when the same allergen enters the body again, the allergen cross-links two or more IgE molecules that are already bound to the mast cell surface. The cross-linking of IgE antibodies triggers mast cell degranulation. I'll show you a picture of what this is. You're never gonna forget the picture. So this right here, this is a mast cell. Okay, so let's say this yellow thing right here is bee sting venom. So in the beginning, somebody gets stung by a bee, and then that person's body makes IgE, and this IgE is specific to this bee sting venom. Once the body makes this IgE comes and binds. It literally comes and binds to this mast cell, right? So the body is it's preparing for attack in the future if this same person gets stung by a bee again. Okay, now later the person gets stung by a bee again. So that bee venom comes and lands on top of this IgE, right? It lands on top of this IgE, and this IgE goes and makes friends with this other IgE and this other IgE, so they link together. This is known as cross-linking of IgEs. And then once the IgEs cross-links on top of the mast cell, they all notice they all have this B venom um, on top of the IgEs, which is the Fab part of the IgE. Okay, now they have the antigen on top of the IgE, and then the IgE all huddled together, also known as cross-linking, and this will cause the mast cell to degranulate, releasing this histamine. So this is degranulation right here, and then it will release histamine, and that's what causes anaphylaxis. Okay, remember this picture, keep it in your head, do not forget it. Late phase of type 1 hypersensitivity. This later phase will happen 4 to 24 hours later and involves mast cell release, like mast cell releasing leukotrienes and cytokines. Okay, okay. So see this. Here we said um, type 1. Usually they do a test, skin test for this, or a blood test for allergen specific IgE. Basically, they're trying to find out if you're allergic to a specific allergen, whether that be eggs, peanuts, or bees, or some type of lotion. Right? I have a cousin who she's literally allergic to almost all cosmetics. So they actually had to do some kind of test for her. I think they did a skin test for her. But that's very interesting because I asked her at the time, I said, "Hey, how do they determine what kind of allergy you have? I mean, you're allergic to everything." She goes, "Oh, they did a skin test on me." I'm like, "Oh." And I don't know if they did a blood test, but we're gonna find out how that's done today. So let's see. Eliza, ELISA is a blood test. It is done to detect specific antigen or antibody, uh, so that ELISA can detect the IgE. Okay, so how this is done? The ELISA test plate, ELISA test. The test plate is coated with allergen. Let's say it's a peanut. The patient's blood is added. If the blood contains IgE against that allergen, the IgE will stick to the plate. A special antibody is then added that binds to human IgE and has an enzyme on it. Okay. So basically, the test plate is coated with peanuts to see if the person is allergic to peanuts, and then you add the patient's blood to it, and if the blood contains IgE against the peanuts, meaning that they're actually allergic to peanuts, the IgE will stick to the plate. So the patient's IgE will stick to the plate. And then another special antibody is added to that human IgE that's stuck to the plate. And that special antibody has an enzyme on it. A color changing chemical is added. If the allergen specific IgE is present, the enzyme turns the solution a yellow color. More color means more allergen specific IgE in the blood. That's interesting. I don't know if I ask you this, but it's like very like I guess you have to know if you're a microbiologist or something. But I mean, it's good to know. It makes more sense. Like, you can connect the dots eventually. How I would summarize this is that to find out if somebody's allergic to peanuts, first you coat the test plate with the peanut, and then you add the human's blood onto it, and human blood, it will have antibodies, and so the IgE in the human blood will stick to the plate if that patient is allergic to peanuts. And then a special antibody is added that binds to human IgE and has an enzyme on it. So basically, first you see if the patient's blood will stick to the um, peanut coated plate, and if it does, you add another antibody that binds to the IgE already on the plate, and then this special antibody has some kind of enzyme on it, um, and then you add a color changing chemical to that test plate, and if the patient is actually allergic to peanuts, the plate will turn yellow. And then the more yellow gets, the more IgE, the, the more IgE specific to peanuts that's in the blood. I ever wonder how people figure this stuff out. Okay, next. Type 2 hypersensitivity. Antibodies bind to cell surface antigens, and this causes cellular destruction, inflammation, and cellular dysfunction. Cellular destruction, the cell is opsonized, means it's coated by antibodies, leading to either phagocytosis and/or activation of complement system, or you have natural killer cells killing using antibody-dependent cellular toxicity. My goodness. You have the natural killer cells killing using the antibody-dependent cellular cytotoxicity. Next, you have inflammation, which is binding of antibodies to cell surfaces. Then you have activation of complement system and Fc receptor mediated inflammation. Cellular dysfunction. Antibodies bind to cell surface receptors. This leads to abnormal blockade or activation of downstream process. Direct Coombs test detects antibodies attached directly to the RBC surface. Indirect Coombs test detects presence of unbound antibodies in the serum. Examples of type 2 hypersensitivity would be autoimmune hemolytic anemia and immune thrombocytopenia. 3. Transfusion reactions. 4. Hemolytic disease of the newborn. Examples of type 2 hypersensitivity would be um, oh, I see. They're all examples of type 2 hypersensitivity reaction, but they're saying this autoimmune hemolytic anemia, immune thrombocytopenia, and transfusion reactions and hemolytic disease of the newborn are all examples of cellular destruction. And Goodpasture syndrome, rheumatic fever, and hyperacute transplant rejection are all examples of inflammation. And number 3, myasthenia gravis, Graves' disease, and pemphigus vulgaris are all examples of cellular dysfunction. Okay, we're going to get into this. So, in type 2 hypersensitivity, antibodies bind to cell surface antigens. This causes cellular destruction, inflammation, and cellular dysfunction. Cellular destruction, the cell is opsonized, which means it's coated by IgG, leading to either phagocytosis and/or activation of the complement system, or natural killer cell killing through antibody-dependent cellular cytotoxicity. Inflammation is when there is binding of antibodies to cell surfaces. This leads to activation of complement system and Fc receptor-mediated inflammation. In cellular dysfunction, we have antibodies binding to cell surface receptors, and this causes abnormal blockade or activation of downstream process. Cellular destruction, you see examples. You see the examples here: autoimmune hemolytic anemia, immune thrombocytopenia, transfusion reactions, and hemolytic disease of the newborn. Okay, so that means that the antigen is attached to the cell and comes to bind. It means that the antigen is attached to the cell and the antibody comes to bind this and causes cell destruction. So basically, um, uh, the antigen is attached to the cell and then the antibody will come to uh, to completely cover that allergen. Allergen or yeah, allergen. And this will cause cell destruction. So first, you have a cell, and then you have, let's say, bee venom. You have the bee venom. This is the antigen, right? So that bee venom is attaching to the cell, and now this activates antibodies. Antibodies will come and wrap around um, the bee venom, cover the bee venom. That is opsonization. And once this happens, once this IgG, so you have it is IgG because we're talking about opsonization and complement. So in type 2 hypersensitivity, you're talking about IgG, um, and in type 1 hypersensitivity, you're talking about IgE only. Just because it's hypersensitivity does not automatically mean that you're talking about IgE. It depends on the type of hypersensitivity. The type 1 hypersensitivity only deals with IgE. Type 2 hypersensitivity deals with primarily IgG or IgM, but we're going to talk about IgG for now. So, yes, once the antigen is attached to the cell, this calls in the IgGs to come and wrap around uh, the B venom, which is antigen, and then this leads to phagocytosis or activation of the complement system, or it activates the natural killer cells to kill, using the CD16 and release granzymes. So here is what it does. So this ADCC, which is antibody-dependent cellular cytotoxicity. NK cells bind IgG Fc region. So the CD16 on the NK cells bind the Fc region of IgG. Okay, so if there's CD16 on the NK cells, the IgG comes and binds to the CD16, but the Fc region, okay, which is the butt of the IgG. Okay, next. Okay, and examples of the cellular destruction would be um, all this, but I want to talk about the hemolytic disease of the newborn. Okay, hemolytic disease of the newborn is very high yield. Um, this is when the mother is Rh negative. So this only happens if the mother is Rh negative, or any kind of negative, O minus, A minus, B minus, AB minus, right? So hemolytic disease of the newborn only happens if the mother is Rh negative. Then she normally has no Rh antibody in the plasma. When she gets pregnant with a Rh positive baby, the Rh antigen on the baby's RBC will mix with the mom's blood during delivery. So now the mom has to make antibodies against the baby's Rh positive antigen. So the mother will now have Rh antibody in her blood, but at this moment the mother is fine because sensitization takes time to happen. That's why this is only problematic for the second baby. If the mom gets pregnant again, if the mom gets pregnant again with a Rh positive baby, then the mom's Rh antibody will come through the placenta and attack the baby's Rh positive antigen and kill the baby. So that's why you have to remember this. And if you guys want to know about blood typing, quickly, this is what it is. This is because once you understand blood typing, there's Rh. Okay. This hemolytic disease in newborn. It will make more sense. So basically, blood typing goes like this: if you are A positive, you have A antigen and Rh antigen on the cell membrane. If you're A negative, you only have A antigen. If you're AB positive, you have both A antigen, B antigen, and Rh antigen. If you are AB minus, you have both A antigen and B antigen, but no Rh antigen. If you're O positive, you have O antigen and Rh antigen. O means you don't have A or B antigen. If the blood type if blood type is positive, it means you have Rh antigen on the cell membrane. If blood type is negative, it means you don't have Rh antigen. That's the primary point. O, if like not just O, any for any of the blood types, A, B, O. Um, what matters if you're positive or negative. A positive blood type means you have an Rh antigen on your red blood cells. If the blood type is negative, it means you don't have the Rh antigen. You do not have the Rh antigen. So this is why the this is what the problem is. The problem is Rh antigen. The problem in hemolytic disease of the newborn is Rh antigen. So um, if the mother is Rh negative, right? So she's Rh negative. She doesn't that okay. So she's Rh negative, which means she's either O minus, A minus, B minus, A, B minus, right? She's a negative blood group. So I'm a negative blood group. I'm O minus. So that means I don't have any Rh antigen. So if I don't have any Rh antigen, I cannot have a uh, yeah. So if I am Rh negative, I cannot have an you know I cannot have a Rh positive antigen uh, or Rh positive anything coming into my blood because that's going to mix with my blood and negative and positive just can't mix together. So what happens is, since I am O negative, that means I don't have a Rh antigen. And let's say my first baby is A positive. That means that baby has an A antigen and a Rh antigen on her cell membrane. Uh, so when she has a Rh antigen on her cell membrane, my body will recognize that as foreign, and my body will develop or make Rh antigen antibody. So antibody will make it will make my body will make Rh antibody against this Rh antigen. but I'm still fine with my first baby because my body takes time to. Um, this is called. It takes time to sense, have like undergo sensitization. So when I have the second baby, and that baby is also A positive, that means that baby has a Rh antigen. And then since I already have this Rh antibody from my first baby, or it's already circulating in my blood, that Rh antibody is going to go and attack this second baby's Rh positive, Rh, yeah, Rh positive antigen. And it's going to kill the second baby. So this my Rh antibody will go through my placenta and attack the second baby's Rh positive antigen and kill the baby. So that's why you know these mothers that have a negative blood type, um, they have to take a shot after the first baby is born and before the second baby is born. So okay, now autoimmune hemolytic anemia is when RBCs are destroyed because antibodies. Okay, it's when the RBCs are destroyed because of antibodies against the RBC. Immune thrombocytopenia is antibodies against platelets. So you can see it's antibodies against. It's antibodies against the cell surface of another cell. Okay. So we have direct Mus test. It's a test that detects antibodies attached directly to the RBC surface. Indirect Mus test is a test that detects the presence of unbound antibodies in the serum. So indirect Mus test is it detects the antibodies in the floating around in the blood, and the direct Mus test detects antibodies directly attached to the RBC surface. Now next we have inflammation. This is binding of antibodies to cell surfaces, which leads to activation of complement system, and Fc receptor mediated inflammation. So here the antibody binds antigen on the cell surface. Yeah, here the antibody binds antigen on cell surface. Yeah, the antibody binds the antigen on the cell surface. But this antibody binding causes the complement system to be activated, and if the complement system is activated, you have neutrophils called in, causing inflammation. So, because complement is activated and Fc receptors are engaged, this calls the neutrophils and leads to um, inflammation and tissue damage. And examples of this would be Goodpasture syndrome, rheumatic fever, and hyperacute transplant rejection. We're going to get into this soon. Okay, next. The third type of third type of type 2 hypersensitivity would be cellular dysfunction. Antibodies bind to cell surface receptors. This leads to abnormal blockade or activation of downstream process. This means antibodies bind to cells that they think are foreign, but those cells are actually part of the body. So the body thinks these cells, um, these receptors on top of the cells are antigens, but it's actually body's own DNA and organs. So this entire cellular dysfunction, um, it's a dysfunction of the receptors leading to decreased or increased activation of receptors. Um, so examples of this cellular dysfunction would be myasthenia gravis, Graves' disease, and pemphigus vulgaris. Pemphigus vulgaris, the antibody will bind to desmosomes, so desmosomes will not attach to cells. So the surrounding cells cannot attach, and this causes increased fluid between cells. This causes bullae and is increased in interstitial fluid. Yes, so pemphigus vulgaris, the antibody will bind to the desmosomes, so desmosomes will not attach to the cells. The surrounding cells cannot attach, and this causes increased fluid because between the cells, and this causes increased bullae. This causes bullae, and there's an increase in interstitial fluid in pemphigus vulgaris. Mhm. Type 3 hypersensitivity, you have immune complex antigen antibody, mostly IgG complexes activate complement, which attracts neutrophils. Neutrophils release lysosomal enzyme, can be associated with vasculitis and systemic manifestations. Serum sickness, the prototypic immune complex disease. Antibodies to foreign proteins are produced, and 1 to 2 weeks later, antibody antigen complexes form and deposit in tissues. This leads to complement activation, and this leads to inflammation and tissue damage. Arthus reaction, a local subacute immune complex mediated hypersensitivity reaction. Intradermal injection of antigen into a pre-sensitized has a circulating IgG individual leads to immune complex formation in the skin. Enhanced local reaction to a booster vaccine characterized by edema, necrosis, and activation of complement. In type 3 reaction, imagine an immune complex as three things stuck together: antigen, antibody, and complement. Examples: SLE, polyarteritis nodosa, host streptococcal glomerulonephritis. Uh, so, okay, so when antigen binds to an antibody floating around in the blood, these antigen antibody complexes accumulate in different parts of the body. This can give activation of complement in that part of the body. This can give you inflammation in that part of the body. Fc receptor antibody will activate C1 complement, and C1 complement will activate whole complement cascade. This will call neutrophils in by C5a. So we're talking. This is what happens in type 3 hypersensitivity. I'm gonna repeat it. When antigen binds to antibody floating around in the blood, these antigen antibody complexes
accumulate in different parts of the body, like in your joints, in your kidneys, uh, in your skin. This can give activation of complement in that part of the body, whether it's skin, kidneys, or your joints. And this will give you inflammation in that part of the body. And so, how does it give you inflammation? The Fc receptor of the antibody will activate C1 complement, and C1 complement will activate whole complement cascade, and this will call neutrophils in by using C5a. Because remember, C5a calls neutrophils in. What else calls neutrophils in? The other neutrophil chemoattractants are, besides C5a, it is LTB4, IL-8, and bacterial products. Now, if I asked you C5a, if C5a was a neutrophil chemoattractant, what would you say? Say no. The answer is no. So see this. C3a is an anaphylatoxin. It increases vascular permeability, mast cell degranulation, leads to histamine release, and inflammation. So C3a is what's involved in type 1 hypersensitivity, because C3a deals with anaphylaxis. And C5a, I know it says anaphylatoxin here. It is, but for step 1, it's usually talking about calling neutrophils in. So you have to remember C3a is for inflammation by mast cells, histamine, vasodilation. C5a is to attract neutrophils. Okay? So remember, C3b is for opsonization, C3a, C4a, and C5a are all for anaphylaxis, and C5a is for neutrophil chemotaxis. And C5b to 9, which is the MAC complex, is for cytolysis. So if I were to ask what calls neutrophils in, you would say C5a. So that would be C5a, IL-8, bacteria products, and LTB4. Okay. If I were to say which were the okay, I'm just going to say which one causes anaphylaxis, you would say C3a, C4a, and C5a. Okay, so they all have A in it, so you remember anaphylaxis, okay? And for neutrophils, you remember C5a because neutrophils typically have around 5 lobes. So remember C5a for neutrophil chemotaxis. And C5b to 9 is MAC complex. For some reason, I just remember that one. I don't know why. C5b to 9. But if you guys are familiar with Mac StudioFix foundations, their StudioFix foundations are called like NC42, NC44, NC45, and they have some they have like different numbers. They have letters and numbers together. So if you remember, if you guys know what Mac foundations are, just remember like it's similar to that. So C3B to 9. Sounds like NC44, right? Okay. So C3b is for opsonization, and then C5a, C4a, so C3b is for opsonization, C3a, C4a, C5a are all for anaphylaxis. So anything that has the letter A in it when it comes to complement, think anaphylaxis for step 1. Okay, so C5a is both for anaphylaxis and for neutrophil chemotaxis, but Neutral chemotaxis. Just remember that for C5A. Okay, next. So normally antigen is from the outside, and our body makes antigen against. So normally antigen is from outside, and our body makes antibodies, not antigen, makes antibodies against that antigen, like in serum sickness. So that's exactly what happens in serum sickness. Serum sickness. What happens first is that the antibodies to antigen are formed first, and then antigen and then antigen antibody complexes form, and then these antigen antibody complexes deposit in the tissue, causing complement activation and then inflammation. So, first, what happens? You have antibodies against whatever antigen you're infected with, and then you have antigen antibody complex that forms, and these antigen antibody complexes go and deposit in your kidneys, your skin, your joints, and then you have complement activation, and then you have inflammation. So what's happening here? These like fever, urticaria, arthralgia, proteinuria, lymphadenopathy. These are all symptoms of serum sickness, and these symptoms happen around 2 weeks after you first get exposed to the antigen. Serum sickness-like reactions are associated with some drugs, may act as haptens. Haptens are a drug that will activate antibody in the body. Okay, so serum sickness-like reactions are associated with some drugs. So if you get reaction to penicillin or haptens, which is a drug that will activate antibody in the body, is a drug that will activate the antibody in the body, you can get a serum sickness-like reaction. Okay. Now, here it says urticaria. Urticaria is when antigen, like urticaria from serum sickness, is when antigen antibody complexes deposit in the skin, and then you have urticaria. Now, proteinuria is when these antigen antibody complexes also deposit in the kidneys, causing proteinuria, and then these immune complexes also deposit in your joints, causing arthralgia. Now, you see here examples of type 3 hypersensitivity would be would be serum sickness, also SLE. What is SLE? SLE in SLE, our own body recognizes our own DNA as antigen. That's the problem. In SLE, our body attacks itself because they're not able to recognize their own. Okay, now that's SLE. Now we have polyarthritis nodosa, which is another example of type 3 hypersensitivity, and this is when you have antigen antibody complexes deposited in the medium vessels, in your renal and GAT vessels. And post-streptococcal glomerulonephritis is also another example of type 3 hypersensitivity. Now you have Arthur's reaction. It's a local subacute immune complex mediated hypersensitivity reaction. Intradermal injection of antigen into a pre-sensitized individual leads to immune complex formation in the skin. Enhanced local reaction to a booster vaccination characterized by edema, necrosis, and activation of complement. So this guy has been exposed to these antigens before the booster. When he first got injection, he got exposed. So this is when somebody gets an injection of an antigen into someone who already has circulating IgG. So I know that was worded quite strangely, but basically, when I said this guy has been exposed to these antigens before, when he first got injection, he got exposed. So this person with the arthritis reaction already has the person with the arthritis reaction already been exposed to a antigen through a vaccine. So whatever vaccine it was, they require a booster shot. So when he got the booster shot, that's when the arthritis reaction happens. So previously, when he got the vaccine, his initial vaccine, um, the body made IgG antibodies against that vaccine. Okay, and the booster shot typically happens a few months later, right? So at that point, this patient already had IgG antibodies in his blood, and when he got the booster shot, what's a booster shot? A booster shot is another vaccine to increase immunity. So whether it be alive or dead, you're still injecting some kind of toxin or bacteria or virus into your body. So that's an antigen. So this booster shot is an antigen, and that antigen will mix with the IgG that's already in his blood from the first shot. Um, and then when the IgG and the booster shot antigen mix together, you get something known as Arthur's reaction. So that's what I meant by this guy has been exposed to these antigens before a booster. When he first got injected, he got exposed, and the booster shot is what makes the reaction an Arthur's reaction. So after booster vaccine, patient develops a painful red swollen area at the injection site 4 to 12 hours later, and that is due to immune complex deposition in the skin. Uh, right. The immune complex deposition means antigen antibody deposition. The antigen here is from the booster shot. The antibody here is from the IgG that was already circulating in the blood. Okay. Um, and so here, when you have a booster shot, um, that's when the antigen and antibody mix together and will cause inflammation by activating the complement. Okay, never done with that. Next, ooh, goodness. Now we're going to go into type 4 hypersensitivity. Okay, type 4 hypersensitivity has two mechanisms, each involving T cells. The first one is direct cell cytotoxicity. Here you have CD8 positive cytotoxic T cell. Okay, here you have CD8 positive cytotoxic T cells kill targeted cells. And the second mechanism seen in type 4 hypersensitivity would be inflammatory reaction. Here you have effector CD4 positive T cells recognize the antigen and release inflammation inducing cytokines shown in the illustration. Okay. Response: does not involve, okay, response does not involve antibodies versus types 1, 2, and 3. Examples is contact dermatitis like poison ivy, nickel allergy, and graft versus host disease. Tests, you have PPD for TB infection, a patch test for contact dermatitis, candida skin test for T cell immune function. The four T's here would be T cells, transplant rejections, TB skin dates, and touching for contact dermatitis. So, type 4 hypersensitivity is delayed, right? It's a delayed reaction. Okay. So, you have to know that direct cell toxicity is when the CD8 positive cytotoxic T cell kills other cells by secreting perforin and enzymes. And the inflammatory reaction, where first it talks about effector CD4 positive T cells recognize antigen and release inflammation inducing cytokines. This is talking about the CD4 positive T cell differentiating into Th1 and the Th1 cells calling in more macrophages to fight. Now I'm going to say that in more detail because we already said it back in the immuno section, but I'm going to say it again. Okay, so they're saying type 4 sensitivity works either through CD8 positive T cells or CD4 positive T cells. Specifically, in CD4 positive T cells, they're talking about Th1 only, because that's the one that increases macrophages. Now, we already know about CD8 positive T cell because there's only one mechanism. Um, it activates the enzymes and perforins, but CD4 positive T cells has so many differentiations. But it's okay. There's only like two you really have to know, which is Th1 and Th2. Th1 is for macrophages, Th2 is for B cells. So we're gonna the deals with macrophages. So this is talking about Th1. Okay, so macrophages secrete IL-12, and this causes CD4 positive T cell to differentiate into Th1 cells. Then the Th1 cells secrete interferon gamma, and this strongly activates macrophages. Th1 cells also secrete IL-2 to increase T cell expansion. Now, activated macrophages then release TNF-alpha, interleukin-1, and IL-12. Macrophages make IL-12 to increase the Th1 population. And then Th1 cells make more interferon gamma, which then activates more macrophages. So this is a positive feedback loop. Okay. So the macrophages and the Th1 are both helping each other to stay up in population. So the Th1 secretes interferon gamma, and the macrophages secrete IL-12 to make more Th1. Okay, so remember that. You have to memorize that. There is no other way. If you look at it 10 times, you will get it. Trust me. Okay. So here, type 4 hypersensitivity does not involve any antibodies. It is strictly talking about cell-to-cell attack and involves macrophages only and T cells. like the CD8 T cells. Okay, so examples include contact dermatitis, poison ivy, nickel allergy. Nickel allergy is when there is belly button inflammation. Also, graft versus host disease is under type 4 hypersensitivity. PPD test for TB infection. This is a patch test for contact. Sorry, PPD for TB infection. This is when um, dead TB is injected into your arm, like skin. So, in short, uh, the dead TB is injected into the skin, and this causes CD4 positive T cells to call in more macrophages, and the macrophages increase inflammation and secrete IL-12 to produce more Th1, and then Th1 would secrete interferon gamma to activate the macrophage, and the macrophage will secrete IL-12 to increase Th1. And this is to check if the patient actually has um, their macrophages and CD4 and Th1 cells working. So specifically it looks like this so that it's easier to understand. For the skin PPD test, typically everyone has had some kind of exposure to TB. So you're supposed to have a reaction. So the first thing that happens is injection of PPD antigen to the skin. Then you have antigen presenting cells like macrophages or dendritic cells present the PPD antigen on MHC class 2 to memory CD4 T cells because you've had prior TB exposure. These memory CD4 T cells differentiate into Th1 cells. Th1 cells release interferon gamma, which activates macrophages, and these activated macrophages release TNF alpha and interleukin 1 and interleukin 12. And this interleukin 12 increases Th1 population again so that Th1 population can release interferon gamma to make more macrophages at that PBD site. And because of all this inflammation, that's why you have the redness within 48 to 72 hours. So it takes 48 to 72 hours for this entire process from step 1 to step 5 to happen. So that's why it's a delayed response, and that's why this is type 4 hypersensitivity. Okay, so yes, you do have to know this is dealing with CD4 T cells. Um, so once the PPD is injected, you have macrophages present the PPD on its MHC class 2. So You have macrophages presenting the PPD antigen on its MHC class 2 to memory CD4 T cells. And these memory CD4 T cells differentiate into Th1. The Th1 secretes interferon alpha to activate more macrophages. And these activated macrophages, in return as a thank you, secrete IL-12, which increases the Th1 response. And these macrophages on the side also secrete TNF-alpha and interleukin-1, which causes this inflammation and redness. Okay. And if somebody has this inflammation redness, that's a good sign. That means their immunity is intact, their CD4 T cells are working, their macrophages are working, um, and they have immunity to PPD, to TB. So if they don't, you have to be cautious if you're talking, if if you're dealing with a AIDS patient or any an immunodeficient person or someone who has like SCID, you know, so they won't have any reaction. They won't have any redness here. Okay, next Candida. Okay, Candida skin test is similar. So Candida skin test is to check if T cells are working. If the patient gets induration, T cells are working. If no induration, T cells are not working. So CD4 positive T cell count is very low if the patient has no Candida, it has no reaction to Candida skin test. If they have no redness to the Candida skin test, that means their CD4 T cells are not working. Okay, their macrophage count is low also. Okay, so what happens in this Candida test? Candida antigen is injected intradermally like a PPD. If the patient has normal T cell function, memory Th1 cells will recognize the antigen because we've all been exposed to Candida at one point or another. Our body knows it, you don't know it, but our body knows it. So the person has normal T cell function, memory Th1 cells recognize this Candida antigen. Within 48 to 72 hours, Th1 cells release interferon gamma, which activate the macrophages. This causes local induration and redness, which is a positive Candida skin test, which is a good thing. If patient's skin is not red, you have immunodeficiency, and that's concerning. Okay. So this is what the step-by-step um, mechanism is for Candida skin test. Candida antigen is injected intradermally. Macrophages or dendritic cells, um, like B cells, okay, or dendritic cells, because APCs are dendritic cells, B cells, yeah. So macrophages or dendritic cells at the site present Candida antigens on their MHC class 2, which means on the macrophages or dendritic cells MHC class 2, um, macrophages, okay, well, let's start that over. So Candigen, it's too late for me, I have to go to sleep. Okay, so Candida antigen is injected intradermally. Macrophages or dendritic cells both have MHC class 2 on top of their cells. So macrophages grab the Candida antigen using the macrophage's MHC class 2 to present it to CD4 T cell. Memory CD4 Th1 cells already sensitized from previous Candida exposure recognize the antigen. Important correction. In healthy people, CD4 T cells are already memory Th1 cells. They don't really differentiate here. Okay. Th1 cells secrete interferon gamma, and this activates the macrophages locally. Activated macrophages secrete TNF alpha, interleukin 1. This causes inflammation, redness, and swelling, which is the good thing that's seen in Candida skin test. That means your immunity is working. If it's not, you have an immunodeficiency, and that's concerning. IL-12 is also secreted by macrophages, and this increases Th1 response to make more interferon gamma, which in turn makes more macrophages. Okay? Alright, done. We are done with hypersensitivities. Very high yield for step 1. Next. We'll do that when I wake up. So, blood transfusion reactions. Allergic anaphylactic reaction. Pathogenesis, you have type 1 hypersensitivity reaction against plasma proteins in transfused blood. IgA deficient individuals should receive blood products without IgA. The timing is within minutes to 2 to 3 hours due to release of pre-formed inflammatory mediators and degranulating mast cells. Clinical presentation. Allergies include urticaria, pruritus, anaphylaxis, wheezing, hypotension, respiratory arrest, and shock.
So what's happening here is that the patient has anaphylaxis against proteins in the donor's blood. So if a person with IgA deficiency gets blood from a regular person that has IgA already in their blood, the person receiving the blood transfusion will have anaphylaxis because they don't have IgA in the blood. So within minutes to 2 to 3 hours, due to release of preformed inflammatory mediators in degranulating mast cells. So what that means is you'll see this reaction within minutes to 2 to 3 hours. First, you have IgA deficient patient getting IgA in the blood. So first you have IgA deficient IgA deficient patient getting IgA blood. So the IgA deficient person is getting blood with IgA in it. Next, the body recognizes this new IgA from the donor as foreign, so the body makes antibody against that IgA, and this is called anti-IgA antibody. So think of anti-IgA antibody as a type of IgE. It's not a type of IgE, but think of it as a type of IgE. So what happens after this, after the body makes the anti-IgA antibodies, they go onto the mast cell. These anti-IgA antibodies go onto the mast cell and cause the mast cell to degranulate. Now, normally in this allergic anaphylaxis reaction, IgE count is normal, but there'll be a lot of anti-IgA antibodies. While IgE and anti-IgA antibodies are not the same, um, think that in IgA deficiency, the person gets the patient gets the allergic reaction to IgA, and then the body will produce anti-IgA antibodies, and those anti-IgA antibodies will go to the mast cell and cause the mast cell to degranulate. But remember, the IgE count is normal, and the anti-IgA antibody count is very high. So you see the mast cell here, you see IgE here, and then in parentheses anti-IgA. Remember anti-IgA antibodies. And this is a type 1 hypersensitivity because it happens very, very fast. Okay. Next, we have acute hemolytic transfusion reaction. This is a type 2 hypersensitivity reaction. Typically causes intravascular hemolysis, like an ABO blood group incompatibility. The timing here is during transfusion or within 24 hours due to preformed antibodies. Symptoms include fever, hypotension, tachypnea, tachycardia, flank pain, and hemoglobinuria, and jaundice. Okay, so here it says typically causes intravascular hemolysis. That means the RBC are being cut up in the blood vessel. So if you have the same kind of antigen with the same type of antibody in the same place, you have hemolysis. What that means is